Melvin Calvin in 1960s utilized carbon-14 radioactive labeling in order to elucidate the pathway of dark reaction of photosynthesis. This marked a historical event because after the unfortunate event of Hiroshima and Nagasaki during the end of Second World War, when human beings began to hate the technology of radioactivity, it is Dr. Calvin who proved for the first time that radioactivity can be used for the welfare of humanity. Thereafter, there are several examples. It's a route in most of our biological laboratories in which radio labeling is used in biochemical assay and pathways. What's the relation between the light reaction and dark reaction in photosynthesis? The function of the light reaction is to produce ATP and NADPH plus H plus in order to convert carbon dioxide into carbohydrate. So once this NADPH plus H plus and ATP are available, light is not required again for the conversion of carbon dioxide into carbohydrate. I have labeled three things A, B and C. Can you just find out what are those A and B and C? The Calvin cycle or the dark reactions consist of three stages. Stage 1, fixation of carbon dioxide by Rubisco. Stage 2, reduction using NADPH plus H plus. And stage 3, regeneration of the acceptor of carbon dioxide. Carbon dioxide is accepted by ribulose 1,5 bisphosphate. This ribulose 1,5 bisphosphate is to be regenerated so that again it can accept carbon dioxide.
So let us see the process of dark reaction or light independent reaction in detail. You already may know where the reaction is taking place in the chloroplast. Let us come to the molecular mechanism. Can you see, this is the starting material RUBP, ribulose 1,5 bisphosphate. So how many carbons are there? Can you see here? This is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. So there are 5 carbons in this compound. This RUBP is a 5 carbon compound. It is the starting material. This RUBP is accepting carbon dioxide. You study like this. One RUBP is accepting 1 carbon dioxide. It becomes 3-phosphoglycerate. But 5 carbons are there. You see, look here. 5 carbons are there in RUBP. 1 carbon is there in carbon dioxide. So 5 plus 1, it should be equal to 6. But the resulting compound, the product should contain 6 carbon atoms. But here you see there is a 3-phosphoglycerate. And how many carbons are there in this 3-phosphoglycerate? 1, 2, 3. So 3-phosphoglycerate is a 3 carbon compound. But I told you here there should be a product with 6 carbons. Actually, there is no single product with 6 carbons. Instead, there are 2 molecules each containing 3 carbon. So 5 plus 1 is equal to 3 times 2. There are 2 molecules of 3-phosphoglycerate. So there are 2 molecules of 3-phosphoglycerate. But what is mentioned here is different. Why? They wanted to balance the chemical equation. 3 molecules of RUBP combining with 3 molecules of carbon dioxide. So 3 molecules of RUBP means 3 times 5 is equal to 15 carbon, plus 3 molecules of carbon dioxide means 3 carbon. So 15 plus 3. So here you have a 15 carbon combining with the 3 molecules of carbon dioxide. So naturally there should be totally 15 carbon plus 3 is equal to 18 carbon, and here you can say 3 phosphoglycerate, 6 molecules, 3 into 6 is equal to 18. So number of carbons is balanced. 3 phosphoglycerate is produced, 6 such molecules are produced. So all the 6 molecules together contain 18 carbon atoms. Here you see 15 plus 3 is equal to 18. So the first product is produced, and carboxylase enzyme is there. Ribulose 1,5-bisphosphate carboxylase enzyme is there. Already we found the name of this enzyme in a previous slide also. Okay. You see, Calvin cycle begins with when a carbon atom from carbon dioxide molecule is added to 5 carbon molecule, resulting 6 carbon molecule is unstable, and it is immediately splitting into 3 carbon molecules. Okay. So this is stage number 1. In the next stage, stage number 2, here 3-phosphoglycerate, which is from the previous stage, is accepting ATP and NADPH plus H plus. From where is the ATP produced? From the light reaction. And from where is the NADPH plus H plus produced? From the light reaction. So the products of the light reaction are used in the light independent reaction. Okay. So with their consumption, 3-phosphoglycerate is transformed into glyceraldehyde 3-phosphate. 3-phosphoglycerate is transformed into glyceraldehyde 3-phosphate. This is an aldehyde, this is a glycerate, this is an acid. So acid is converted into aldehyde. Okay. And this glyceraldehyde 3-phosphate, each is consisting of 3 carbon atoms. So there are totally 6 molecules. Remember, balance the equation, we are beginning with here 6 molecules. And glyceraldehyde 3-phosphate is later converted into glucose. You know that glucose consists of 6 carbon. So definitely more than one glyceraldehyde 3-phosphate molecule is required for the formation of glucose. How many carbons are there in glucose? Glucose contains 6 carbons. How many carbons are there in glyceraldehyde 3-phosphate? Contains 3 carbons. Okay. So how many glyceraldehyde 3-phosphates may be required to form one molecule of glucose? You just calculate. It is written here through a series of reactions. Remember, this is not a single step reaction. This is a series of reactions. Energy from the ATP and hydrogen atoms supplied from the NADPH plus H plus are added to the 3 carbon molecule. What is the 3 carbon molecule? 3-phosphoglycerate. The reduced 3 carbon molecule, by accepting hydrogens, it is reduced. The reduced 3 carbon molecules either combine to make glucose, that is what we found just now, or are used to make other molecules that we have not seen. Now, third stage, regeneration. So first one is carboxylation, second one is reduction, and third one is regeneration of RUBP, of the starting material. Regeneration of RUBP from the glyceraldehyde 3-phosphate. This also is consuming ATP. This regeneration also is consuming ATP. Look here. Here, for converting 6 3-phosphoglycerate molecules, we need 6 ATP and 6 NADPH. Here, for regenerating RUBP molecules from glyceraldehyde 3-phosphate, we need a total of 3 ATP. Once again, we need to convert 5 glyceraldehyde 3-phosphate molecules into 3 RUBP molecules. How many carbons are there in 3 RUBP? It is 3 times 5. How many? 15. And how many carbons are there in glyceraldehyde 3-phosphate 5 molecules? Each of them contains 3 carbons. totally 5 molecules are there. So 5 times 3 again is equal to 15 carbons. So RUBP is completely regenerated. I mean 3 RUBP molecules are completely regenerated using 3 ATP. Look here, here we started with the 3 RUBP molecules. Here 3 RUBP molecules are regenerated. So finally RUBP is not consumed. Ribulose 1,5-bisphosphate is not consumed. Why? Whatever RUBP is consumed here is finally regenerated. Okay, make a note of all those things. Plus, how many ATP are totally required for the process? 6 here and 3 here, 9. So when 9 ATP molecules and 6 NADPH plus H plus are consumed, 1 molecule of glucose is produced, we may say. So most of the reduced 3 carbon molecules are used to regenerate the 5 carbon starting material. So the cycle is completed. Here is another diagram in which the same processes are explained. Then what's the difference? In this diagram you can see the enzymes are mentioned clearly. That the first enzyme, the carboxylating enzyme is Rubisco. Ribulose 1,5-bisphosphate carboxylase oxygenase, Rubisco. Second one, it is PGA kinase mainly, and also glyceraldehyde 3-phosphate dehydrogenase, another one. And here is the stage in which glyceraldehyde 3-phosphate is used for making glucose and other sugars. Here the name of the enzyme is not mentioned simply because it consists of many stages. We cannot simply say the name of any one enzyme. And here is the regeneration of RUBP. Try to get more ideas from the textbook. Can you identify from the diagram, from the color of the diagram, from which textbook is it taken? And where is the process taking place? Look here, it is mentioned here very clearly. Again the same process, but here the difference is that the text, whatever I told you earlier, is written here very clearly. CO2 molecules are captured by RUBP, like that the text is explained here, and also the calculation of carbon numbers also is given here very clearly, and NADP, ADP, ATP, etc. are also given. And so this will help you to understand the subject matter in a better manner. Okay. We were talking about Rubisco. Rubisco is a key enzyme of the light independent reactions. When we found the name of Rubisco, it is Ribulose 1,5-bisphosphate carboxylase oxygenase. It means this enzyme can catalyze oxygenation also. It can catalyze oxygenation also, which means this enzyme can add carbon dioxide to RUBP. Then we will call it carboxylase. It can also add oxygen to RUBP. Then we will call it oxygenase. And whenever oxygen is adding, that process is actually called photorespiration. So when is it taking place, this photorespiration? You see, in the atmosphere, the concentration of oxygen is 500 times more that of carbon dioxide. So what happens, there is an equal opportunity that this Rubisco may bind with the oxygen as well, especially when there is a decreased CO2 level due to high rate of carbon fixation in intense light, and also when the temperature increases. These two things, increased light intensity and increased temperature, both exist in the tropical regions of the earth. Therefore, in the tropical regions, in the tropical plants, there is a higher tendency for photorespiration. And, where is this process taking place? Which are the organelles required for this process? Can you see? This is the chloroplast, and near to the chloroplast we have peroxisome, and adjacent with that we have mitochondrion. This chloroplast and peroxisome and mitochondrion, all three organelles are required for photorespiration to take place. How is it proceeding? Rubisco is accepting oxygen. That means RUBP is combining with oxygen. The reaction is catalyzed by Rubisco, and what is produced is 2-phosphoglycolate. Remember, what is the product when carbon dioxide is accepted instead of oxygen? So 2-phosphoglycolate is produced, and where is it taking place? It is taking place here. It is mentioned very clearly. And then it is transformed into glycolate. This glycolate is moving out of the chloroplast and it is entering into peroxisome. In peroxisome, this glycolate is converted into glycosylate, then it is converted into glycine. And this glycine is moving out of peroxisome and it enters into mitochondria, where 2 molecules of glycine are giving rise to 1 molecule of serine. Glycine consists of 2 carbon atoms. Serine consists of only 3 carbon atoms. So 2 molecules of glycine means 4 carbon atoms. So a 4 carbon substrate is giving rise to a 3 carbon product, because one of the carbon is lost as carbon dioxide. This is a precious loss for the plants, because it is very difficult to fix carbon dioxide by means of photosynthesis. That precious carbon is lost in the process. And so here carbon dioxide is lost in that process. Where is it taking place? It is taking place in the mitochondrion. This serine is going out of mitochondrion. It is entering back into peroxisome, where it is converted into glycerate, and from peroxisome it is moving into chloroplast, where it is converted into 3-phosphoglycerate and it is entering into Calvin cycle. So is there any benefit? If you look at this diagram, is there any benefit for the plant because of this photorespiration? Try to understand yourself. It is accepting oxygen and releasing carbon dioxide. So photorespiration is the process in which plants are accepting oxygen and they release carbon dioxide. So in that aspect, it is similar to ordinary respiration. What you see here is actually a micrograph. This is a real picture of the plants, and what is happening during photorespiration is mentioned here. I will take the highlighter so that you can see it better. Now you can see here peroxisome. I told you peroxisome is involved. I told you mitochondrion is involved, and also chloroplasts are involved. So what is happening there? In the previous slide, we found the various stages of photorespiration. You can see here, this is the mitochondrion, this is the mitochondrion. Here the carbon dioxide is going out and the serine is produced. And here is the organelle, which is this organelle. And here you can see the chloroplast also. So chloroplast is involved, peroxisome is involved, mitochondrion is involved. Okay. So this is actually an electron micrograph of the leaf tissue where the process is taking place. And it's given here already. This is from the tobacco plant leaf, showing a peroxisome. Where is the peroxisome? Here identified by the crystalline core. Here you can see it, and close to a mitochondrion. You can see the mitochondrion here. And the reactions of the photorespiration that occur in each of these organelles are shown superimposed. So it's a very good picture for you to understand. You can identify the size of this organelle because you can see the scale is given here. You can use a scale also. This much distance is 0.3 micrometer. So this much is 0.3 micrometer. This much is 0.3 micrometer. Like that, you try to identify what is the size of this these organelles. It will help you a lot.
So far, we were talking about a photosynthetic pathway in which the first product of carboxylation is a 3-carbon compound. We studied RUBP combines with carbon dioxide forming 2 molecules of 3-phosphoglyceraldehyde, and of course this is a 3-carbon compound. And also we found that there is a pathway called photorespiration. It is present in the plants of a tropical region. In this process, the plants are absorbing oxygen and they are releasing carbon dioxide, and this is a waste for the plants. And in order to avoid photorespiration, some types of adaptations have been developed, and a new pathway itself is evolved. We call it C4 pathway. You can see here C4 pathway. What is C3 pathway? C3 pathway is the photosynthesis we studied so far, in which the first product of photosynthesis is a 3-carbon compound. While in the new pathway, which is actually better evolved, we call it C4 pathway simply because the first product of photosynthesis, the first product of carbon fixation will be a 4-carbon compound. First of all, let us see and compare the anatomy of the plant organs involved. See, this is the C3 plant. What you see here, this is the C3 plants. Calvin cycle takes place in mesophyll cells, while the bundle sheath cells are non-photosynthetic. You see, this is the upper epidermis. This is the lower epidermis. You can see the stomatal apparatus here. And in between the two epidermis, whatever you see here is called mesophyll. Whatever we see here is called mesophyll. And in dicotyledons, we may say it is divided into two, or can be distinguished into two, that lower spongy mesophyll and upper palisade mesophyll. Okay, this is the vascular bundle. Do you know what is vascular bundle? The tissue which is consisting of xylem and phloem and which is carrying water, food materials, etc. So here we have the xylem and phloem, and the sheath of cells covering the vascular bundle is called bundle sheath cells. Bundle sheath cells. And this is the C4 plant's anatomy. You have the upper epidermis, you have the lower epidermis. Between them, we can see the complete mesophyll. Remember, it is not differentiated into palisade and spongy. And here you have the xylem and phloem, and here you have the vascular bundle. How do they differ? Point 1, palisade and spongy differentiation is not there in C4 plants, but that is not our point. Our point is that this bundle sheath tissue, bundle sheath cells are photosynthetic in nature in C4 plants. While in C3 plants, bundle sheath cells are not photosynthetic in nature. So in C4 plants, the mesophyll cells are taking part in photosynthesis, and also the bundle sheath cells are taking part in photosynthesis. While in the C3 plants, the mesophyll is taking part in photosynthesis, but the bundle sheath tissue is not taking part in photosynthesis. That is the difference. Let us see how is this process taking place and how this process is going to reduce the rate of photorespiration in C4 plants. And where are the C4 plants actually present? Where photorespiration should be avoided. So where is the photorespiration taking place? In the tropical plants. Therefore, C4 anatomy is present in tropical plants only. For example, you know sugarcane.
Sugarcane is a good example from where you are getting that common sugar which is used in your tea and coffee. The sugarcane is a very good example of C4 plants. So why we call it C4? Because the first product of carbon dioxide fixation is a 4 carbon compound, and that is taking place in the mesophyll cells, where this is the mesophyll. So in this mesophyll cells in the chloroplast, the 4 carbon product is produced. Then these 4 carbon compounds are transferred from the mesophyll to the photosynthetic bundle sheath. So from the mesophyll, the 4 carbon compound is transferred to the bundle sheath, and Calvin cycle is taking place in the bundle sheath cells. In the C3 plants, there is Calvin cycle. In the C4 plants also there is Calvin cycle. Then what's the difference? In C3 plants, both the light reaction and dark reaction are taking place within the same cells. In the C3 anatomy, in the C3 plants, both the light reaction and the dark reaction are taking place in the same cells. While in the case of C4 plants, light reaction is taking place in one type of tissue, while dark reaction is taking place in another type of tissue. So light reaction in which oxygen is evolved or oxygen is produced is spatially separated from dark reaction. So let us see the pathway in detail. So here we are going to see the actual process, which are the cells involved. You can see this is the mesophyll cell, while here is the bundle sheath cell. You can see the boundary of the cells clearly. The cells are interconnected through connecting pathways. You might have heard of the term plasmodesmata, something like that. They are actually connections through the adjacent cells. So where is carboxylation taking place? You can see here PEP, phosphoenol pyruvate. It is a 3 carbon compound. It is the acceptor of carbon dioxide, not RUBP. So a 3 carbon PEP is accepting carbon dioxide. So 3 carbons here plus 1 carbon here, we get a 4 carbon compound. The name of the 4 carbon compound is oxaloacetate. Oxaloacetate. Where is it taking place? In the mesophyll cell. And what's the name of the enzyme? Phosphoenol pyruvate carboxylase. PEP carboxylase is the name of the enzyme responsible for carboxylation. Later, in the mesophyll cell itself, this oxaloacetate is isomerized into malate. Isomerized, they are isomers. And this malate is transported through the plasmodesmata into the adjacent bundle sheath cell. In the bundle sheath cell, you can see here, malate is regenerating pyruvate. Malate is a 4 carbon compound. Pyruvate is a 3 carbon compound. So the 4th carbon is released as carbon dioxide, where in the bundle sheath cell. And here in the bundle sheath cell, Rubisco is there, RUBP is there. That RUBP combines with the carbon dioxide. The reaction is catalyzed by Rubisco, and a normal Calvin cycle is taking place, where in the bundle sheath cell. While this pyruvate produced here moves back to the mesophyll cell, and using ATP, it is becoming phosphoenol pyruvate. So basically what is happening here? This pathway is a carbon dioxide pumping mechanism. Carbon dioxide is coupled with the PEP to form oxaloacetate. It is transformed into malate, and this malate is transported into bundle sheath cell, where carbon dioxide is released. So this is a mechanism to increase the concentration of carbon dioxide around Rubisco. A mechanism to increase the concentration of carbon dioxide around glucose, such that, not glucose, around Rubisco, to increase the concentration of carbon dioxide around Rubisco, such that Rubisco does not get a chance to combine with oxygen. and light reaction is not taking place here. In this bundle sheath cell, light reaction is not taking place. Therefore, oxygen is not evolved. Therefore, no oxygen comes there to compete with the carbon dioxide for the active site of Rubisco. Then you have a question. Where is the light reaction taking place? From where is the NADPH plus H plus and the ATP required for the dark reaction produced? Those things, the light reaction also is taking place in the mesophyll cell. It is not mentioned here. In this diagram, it is not mentioned. But the light reaction is taking place in the mesophyll. Water is split up there. Oxygen is released there. ATP and NADPH plus H plus are produced there. And that ATP and NADPH plus H plus are also brought to the bundle sheath cell. Those things are not mentioned in this diagram, in this picture, but we will see it later.
Here you can see the chemical reactions taking place during C4 pathway. Where do we start? Where there is carboxylation. Look here, carbon dioxide is reacting with PEP to form oxaloacetate. But what you see here is something different. How is it? You see, wherever carbon dioxide reaction is involved in living system, the carbon dioxide first dissolves in water to form carbonic acid. The carbonic acid dissociates into bicarbonate ions, and those bicarbonate ions are reacting with the phosphoenol pyruvate. Always that is the case. So this is the new coming carbon, and using NADPH plus H plus, it is reduced to malate. Malate is transported to bundle sheath. There, this carbon dioxide is released. Pyruvate is also released. You see here, this new coming carbon is given in the red color. Here is the carbon, here is the carbon, and here is the carbon released. So the carbons which are reacting with the PEP is ultimately released in the bundle sheath cell. And of course, pyruvate is produced and it is transported, and the PEP is generated utilizing ATP. So you remember one thing, one molecule of NADPH plus H plus is consumed here, but that NADPH plus H plus is regenerated here. So there is no expenditure of NADPH plus H plus in this process. Secondly, ATP is consumed. How? One ATP is giving rise to AMP, because two high energy bonds are broken, equivalent to two ATP molecules, we should say. Two high energy bonds are consumed here. We should keep that in mind. This carbon dioxide pumping mechanism requires energy. What you see here is a real electron micrograph of the leaf cells. The reactions taking place are overlaid in that diagram. That reactions taking place are overlaid in that diagram. For example, you can see here, this is the mesophyll, this is the bundle sheath, here is that plasmodesmatal aperture, that opening, and carbon dioxide is taken from the atmosphere, it is got transformed into oxaloacetate and malate, it is transported. So the entire pathway can be seen here. Remember, and here you can see, measure it using a scale, because this much distance is 5 micrometer. This is a very good diagram taken from a textbook. Can you identify from which textbook is the diagram taken? Like this, you should refer different books to get more ideas into the matter we are studying. This is a very good diagram for you. Now we are going to a related topic, crassulation acid metabolism, CAM. Why is it essential? I told you, in the tropical regions, there is the problem of photorespiration. This crassulation acid metabolism, CAM, is another strategy in some plants to minimize the occurrence of photorespiration.
in very hot regions, if the plants open their stomata during their daytime to take in carbon dioxide, you know what will happen? They take in oxygen also. It will lead to photorespiration. Also, they may lose a lot of water by means of transpiration. Both these are harmful because in the hot regions, water is a limiting factor and the precious water cannot be lost simply by evaporation. So they have solved these problems by opening their stomata during night. You see, a second strategy to decrease photorespiration in hot regions. It is taking place in succulent plants, for example, cactus, pineapple, etc. You are very much familiar with the pineapple, and we call it crassulation acid metabolism because it was discovered for the first time in the members of the family Crassulaceae. It's very simple. The stomata, the openings through which the gaseous exchange is taking place, is opened during the night and they are closed during the day. So what happens during the night? They keep their stomata open. They take in carbon dioxide. They carry out a C4 pathway. A C4 acid is produced, and that C4 organic compound is accumulating in that organ throughout the night. By daytime, they close their stomata. Already the C4 acid is stored there, so they close their stomata, and just like in C4 plants, they are releasing their carbon dioxide. They release their carbon dioxide, and an ordinary C3 pathway is taking place. See here, in the daytime, the high levels of CO2 drive the Calvin cycle. This is helpful to reduce photorespiration. Just like in C4 plants, the CAM plants use both C4 and C3 pathway. They differ from C4 plants in that they use C4 pathway at night and the C3 pathway during the daytime. What about the C4 plants? In the C4 plants, both these pathways, C3 and C4 pathways, were taking place during daytime itself, but in different tissues. Here in CAM plants, C3 pathway is taking place during the night time, and C4 pathway is taking place during the in the crassulation acid metabolism plants, C4 pathway is taking place during the night time to accumulate the C4 acid, and C3 pathway is taking place during the daytime. Okay. And it's given here. In these plants, crassulation acid metabolism plants, both C3 and C4 pathway are taking place at different times, day and night, but within the same cells. Make a note of it. Within the same cells. What we see here is a pictorial representation of what we talked in the previous slide. What's the difference between the C4 and C3 plants, and what's the similarity between the C4 and CAM plants? You see, in C4 plants, there is the C4 pathway and C3 pathway, but they take place in different types of cells, but at the same time. And in CAM plants, there is a C4 pathway and there is a C3 pathway, and C4 pathway is taking place during night time, and C3 pathway is taking place during daytime, but both the pathways are taking place in the same cell. While in C4 plants, these are taking place in different cells. So that is the difference. So here are the reactions of crassulation acid metabolism. Basically, reactions are the same as we see in C4 plants. See here, during the night time, I'll take the highlighter. During the night time, when the temperature is lower, stomata are opened, carbon dioxide is taken in, and the carboxylation reaction is taking place, what is produced is, as usual, what is produced is oxaloacetate, and that oxaloacetate is transformed into malate. The malate is stored in vacuum. This process is taking place throughout the daytime, and throughout the daytime, the C4 acid is produced from phosphoenol pyruvate. The C4 acid is produced. Enzyme is phosphoenol pyruvate carboxylase. Oxaloacetate is formed. Malate is formed. The malate is stored in the vacuum throughout the night time. And what happens during the daytime? Daytime temperature is very high. You can see here, daytime temperature is very high. Stomata are closed, so there is no loss of water. And from the malate, from the vacuum, malate is undergoing decomposition to produce carbon dioxide, and the carbon dioxide is used in C3 pathway to produce normally glucose and other metabolic products. So both the reactions are taking place in the same cells. That's the difference. And in the vacuum, malate is stored. That malate is released into the chloroplast, because this tissue is actually the chloroplast. And inside the chloroplast, this reaction is taking place. So both the reactions are taking place in the same tissue. But the difference is that one is taking place during night time at low temperature. The other one is taking place during daytime at higher temperature. This pathway, glycosylate pathway, is taking place only in plants and not in animals. What's the importance of the pathway? You can see here that the starting material is fatty acid, or we say lipids. The starting material is fatty acid. It is entering into, it is broken down by means of beta oxidation to produce acetyl-CoA. It is entering into citric acid cycle, just 2 reactions only. It is transformed into succinate. Thereafter, what happens? This succinate is exported from the glyoxysome to the mitochondria. So, for this pathway, glyoxysome is required and mitochondrion is required. So the succinate is exported from the glyoxysome to the mitochondrion. There, this succinate is undergoing some of the reactions of the citric acid cycle. You can see these reactions are those of citric acid cycle. And it is forming oxaloacetate. Thereafter, this oxaloacetate is transformed into PEP, and this PEP is transformed into carbohydrate. Remember, for this purpose, the oxaloacetate is transported out of the mitochondria. So the carbohydrate, sucrose, is formed in the cytosol. So wherever this pathway, glycosylate pathway, is taking place, glyoxysome is involved, mitochondrion is involved, cytoplasm is involved. What's the advantage? You know that in many plants, reserve food material is lipid. For example, in coconut, reserve food material is lipid. And this reserve food is used during germination so that the germination seedling can obtain energy. And the immediate source of energy is carbohydrate. You know that when a coconut is germinating, that white structure developed there is quite sugary in nature. It is very sweet also. It's because this pathway is taking place there. So lipid can be transformed into carbohydrates. This enzyme is some of the enzymes of the pathway are not there. Can you find out which of these enzymes are not there in our body? These enzymes are there in our body. These enzymes are also there. These enzymes are there. Then can you find out which enzyme of this pathway is not present in our body? Because of that deficiency only, we cannot carry out this pathway. Okay, this is a wonderful pathway by which plants are converting lipid into carbohydrate.